A very good afternoon to everyone who's turned into this live session. My name is Kadambiri, and on behalf of Team RoverX, I welcome you all to today's live session where we delve a little into understanding the unknown. No, this isn't about the aliens or the wow signals. So, this session, we will explore our own solar system and what makes it such an interesting place to search for extraterrestrial life. Since NASA's publication on astrobiology strategy in 2015, significant scientific, technological, and programmatic advances in the quest for life beyond the Earth have taken place. In today's session, we'll look into what astrobiology missions are and why they are so important in the further development of space exploration programs and for the future human civilization in general. Before we get into this session, I would encourage you all to be interactive and feel free to type in your doubts or questions in the live chat. Thank you.
Thank you, Kandabari. Now it is my pleasure to welcome and introduce you to our guest speaker, Dr. Siddharth Pandey. Good afternoon, sir. Dr. Pandey is the head of Center of Excellence in Astrobiology at Amity University, India. As a part of this, he is leading an initiative to establish India's first dedicated center for astrobiology and space biology research in the country. Dr. Pandey is a space engineer with project management and leadership experience and has worked with various teams of astrobiologists. Prior to this, he has also experience in building sample collection instruments for Mars and Venus surface missions while working at NASA Ames Research Center. He has been engaged in education and public outreach activities and is motivated to use space as a tool to spread awareness, social consciousness, and inclusiveness within our communities. He holds a PhD degree in aerospace engineering from UNSW Canberra, Australia, MS in space systems engineering from TU Delft, Netherlands, and BTech in aerospace engineering from Amity University, India. Sir, it is truly an honor to have you here with us. And now I would request you to kindly take over and start the session.
Thank you so much, Vikyat and Kadambari. Greetings to everybody joining in. It's an absolute pleasure and honor to talk to VIT University. And I'm really thrilled to interact with the team RoverX. And I hope through this session, I can bring forward some aspects of astrobiology that could be incorporated by students in their day-to-day life, in their upcoming professions, or perhaps even dinner table conversations, depending on how serious you are about this topic. This is up and around you. You cannot avoid it. It's there. It's all happening. And hopefully, with a few slides, I would have given you a presentation of my thoughts into this story, as well as talk about certain aspects of astrobiology that might be of interest to all of you. All right. So, can someone confirm my screen is sharing?
Yes, sir.
Okay. Thank you. Right. So, here's what we're going to do. I'll talk about astrobiology from the perspective of space missions, right? So we all know that we have been looking for life now for almost or more than 3 decades. But why is it really so hard to look for life? And I'm going to be focusing on our solar system and the different worlds that exist in our solar system and talk about how hard it is, how challenging it is, and why is it so hard. And hopefully, some of those challenges might be things that you might find interesting enough to consider, tackling, so that perhaps the search for life could in fact be fastened by some efforts coming in from students listening in, just like yourselves. I will also talk about my center towards the end of the talk and certain existing projects and opportunities that are there for students to undertake either summer internships or research projects or even perhaps full-time opportunities, depending on how we go. We partner with several national and international organizations, and we are very fortunate to get their support to be able to do this exciting piece of research, which is the first ever such center in the country that is solely dedicated for astrobiology. So I'm very, very honored to be able to talk to you about this field, and I hope to get a lot of enthusiasm in return from everybody else who's interested in it. So space missions in the search of life, right? Now, there might be a few of you who might not have heard this word astrobiology before, or might have heard it and might not be able to immediately place a definition towards it. So let me start with giving you what astrobiology is defined as conventionally by a few of us. Astrobiology pertains with the study of the origin, the evolution, the distribution, and the search for life in the universe. So it tackles all these four different words that I mentioned: origin, the evolution, the distribution, and the search for life. These four main keywords in the definition of astrobiology. And it's a very interdisciplinary field. Well, our focus immediately goes towards biology as soon as we read astrobiology, but it's actually a lot more than biology. It has a lot more to do with the environment in which this biology could have or could have not formed. And what does that environment undertake? It undertakes the landform or the landscape, and that's where the geology aspect comes in. So it's important for geologists to be aware of. It's important for astrobiologists to be aware of geology. What else comes in the environment? You have different kinds of atmospheres. You have any liquid bodies like water, rainfall, winds. So there's all these kind of different atmospheric sciences that are also relevant, different kinds of chemistries that are involved, because at the end of the day, everything can be broken down into molecules, right, and elements. So the chemistry aspect is also important. And if you're looking at how planets form, planetary science as a field becomes important to understand. You talk about gravity playing an important role as well in the formation of different bodies, and then eventually the conditions in which life might have started. So physics is again very important. And of course, when you're trying to reach these worlds to undertake all of these missions, you have engineering, wherein you have to undertake complex technical operations, and that's where the engineering aspect comes in to be able to fulfill your mission. So astrobiology is not just people who do biology. It's engineers, it's geologists, it's chemists, physicists, it's even philosophers. When we talk about different levels of thought that go in, when you're trying to develop the logic, the rationale, and the hypothesis building that is involved when you're trying to ponder about whether life could have started or could not have started. So it's a heavily interdisciplinary field, and that's something that invites collaboration. It invites discussions, and that's the best part in my mind about this field, that it brings people together to be able to work towards answering a common question. So in today's talk, I'll be covering what are the different missions that are undertaken for astrobiology, and I'll be focusing on our own solar system. And the main thing that I would like to point out are the unique challenges that are faced by them. Why is it so hard to look for life? And eventually, what are the consequences of these results? We haven't really found our extraterrestrial so far, but there have been several different discoveries along this path. And how do those discoveries add up and shape the upcoming future missions? Now, there's already several lists that kind of put together a table of different missions that have taken place, and one of them can be found on NASA's astrobiology website to kind of give you an idea of the different missions that have taken place so far. So what is it when we say that we're looking for life? What is an astrobiology mission looking for? So it's mainly looking for two things. The first are called ingredients, and the second are called byproducts. So what do we mean by ingredients? Just like when you're going about baking a cake, you are really looking at what is it exactly that goes into making a good cake. So you've got your sugar, you've got your butter, you've got baking soda, you've got chocolate if it's a chocolate cake, and those are the essential initial elements before the cake formation has taken place. So similarly, when you're going to look for life, we know already, based on our understanding of life here on Earth, that there are certain essential elements that you want to look for: carbon, hydrogen, nitrogen, oxygen, phosphorus and sulfur. So these are the main elements, and then you have different molecules, organic molecules that kind of existed before life came into being. So those are the ingredients, and then the byproducts. So after the main living organism has been formed, what is the leftover parts that exist around it? So when we are trying to look for life, even if you're not able to find that living organism or the fossilized structure or any decaying leftover remnant of that living organism, if we can find these ingredients and byproducts, it can help us build the story as to what might have taken place, what kind of biological process might have taken place. So that's the what of it. And the how. how do we go about doing that is mainly in these two areas. These are the primary sources of information, that is either chemical in nature or morphological in nature. By chemical, we are mainly talking about spectrometers, by when we break down the light that is coming back from the source, and based on the wavelength, we are able to then characterize the source of that light and associate that wavelength with a particular signature of a mineral or of a particular gas or a particular element that might be emitting that light. We have gas detectors, we have different forms in which we can conduct chemical assays and confirm whether these organisms are uh, formulated because of a biological source or a non-biological source. So that's our important tool using chemistry. And then the morphological signatures, in which we have the different imaging systems, wherein we are able to absorb this light and look at it across the spectrum, electromagnetic spectrum, and then be able to associate what we are finding with what we see here on Earth. So it could be perhaps a particular large scale image that in which you're looking at some kind of hilltops, you're looking at certain gullies, and you're looking at drainage basins. And then if you're looking at close up images, you're trying to look at associated structures, so microscopic images, then you're zoning in, and then you're focusing in on certain, for example, you're looking at certain fossilized deposits, and you're trying to look at it even more closely and confirm whether these deposits are actually of any remnant biological activity, or it could be something that is just regular geology at play. So both of these are important tools, chemical and morphological signatures. And how do we exactly go about doing this? So we have different kinds of missions. We have missions in which you involve orbiting your target world. It could be around a planet, or it could be around a specific moon, or you're in fact just doing a flyby or a fly through of that. You also have missions that eventually land onto the surface, and which of course are even more exciting because you get up close, in close proximity to these different sites that you would like to study. And eventually you talk about returning the samples back into your laboratories. So for those who are uninitiated into some of these terminologies of flybys and orbits and landings, just put together something very simple. So you have flyby missions in which you fly towards your target world, and then eventually, rather than entering into an orbit around it, you just kind of use its gravitational pull to slingshot around it. And while you're doing that, you get a duration of time in which you are in really close proximity with that particular surface. And while you're there, your instruments go haywire and crazy, and you're able to take a lot of images. You're able to do a lot of measurements of the environment, that the near surface environment, the atmosphere that you might be flying through. And one example of it was the New Horizons mission, which actually did a flyby of Pluto and was able to capture so much of information while it was doing that. And there have been other successful examples as well with Voyager, Galileo, Cassini-Huygens, and Hayabusa missions. The other class of missions are orbiter missions, in which you enter into a stable circular or elliptical orbit around your target world. And in that case, you're able to spend more time, months to over years, and you continue to carry out different observations. It could be based on cameras, or you could constantly be taking certain atmospheric measurements while you're there. And of course, some of the most exciting missions have been the ones that have approached the planet. For example, if it is Mars, entered into the atmosphere, and because you're traveling with such high velocities, you have to break the vehicle, decelerate it in a very short duration of time, within a few minutes, and then eventually have a soft landing on the surface. And the next phase of missions are called the sample return missions, which will eventually be taking samples, lifting off from the surface, and then coming back. Something that has been successfully done from the moon, but not yet from Mars. There are also other missions that look for life outside our solar system, and these are mainly space telescopes missions like Hubble Space Telescope, James Webb, Kepler. But all of these missions are outside the scope of the work that I have been doing so far, but to mention the fact that definitely they do take place. And also around our own planet, we have several experiments that have been conducted on the International Space Station. And these experiments that take place on the International Space Station, many of which are related with studying how living organisms, you know, kind of go about their living in a very unique environment. And what is it that our own low Earth orbit has to offer? You have reduced gravity, you have exposure to radiation from space. So here is an experiment that you see being handled by the astronaut, which is a box full of different plant, fungi, bacteria that was kind of packed up inside these boxes, which actually have a clear glass on the top, as you can see on the lower right. And that unit called expose was actually, and there's been at least 3 different forms of those units that have been installed on the external side of the International Space Station. On the top right, you see the International Space Station, and you see where these different platforms where they have been installed. And they were kept there for a long duration of time to allow space, which is mainly radiation and the temperatures, to actually affect their living, the growth of those organisms. And then they were actually sent back to Earth. They were carefully retracted back into the ISS and then stored away. And then when the astronauts returned, they actually brought them back. And once they were back, they analyzed them and tried to understand how exactly did the space environment affect the growth of living organisms. Now, why is that important to astrobiology? Well, that tells us how do organisms behave in these unique physical chemical conditions. I should also point out that my own center, we have been selected for 2 spaceflight experiments as part of ISRO's PS4 orbital platform. So if this pandemic had not happened, we would have already flown our first payload, but we're still awaiting ISRO's launch schedule, and the payload is back with us. We retrieved it back, and we've made certain modifications to the hardware. And it's mainly a unit that will be studying the growth of plant samples. And as soon as it is up, we will be able to compare it with a similar study that will be taking place in our own space biology lab at Mumbai, and be able to do comparisons. And the second experiment is a more extensive experiment that will be also sent up to the low Earth orbit environment. So let's talk about the different criteria that need to be met by these instruments when they are looking for life. So this is a list that has been put together, and for more information, I have left the link of this paper in Astrobiology journal, published in November of 2018 by Mark Neveu and team. And it talks about these different criteria. So you're talking about the sensitivity of the instrument, the fact that it has to be really, really sensitive to be able to detect very minute traces of certain organics. Secondly, it needs to be contamination free. So you want to ensure that when you're sending something to another planet, it's not actually carrying samples from Earth to there, so that once it powers on there, it's actually not detecting something that's actually flown with it from Earth. The third is repeatability. It has to be a really, really rigorous instrument that is able to measure something and do it again and again to show that it's something that's not just a false positive, it's something that has been actually detected, right? And all of these 3 things were actually met by the first astrobiology mission to Mars, which was the 1976 Viking lander that landed on the surface of Mars. And even though these were successful experiments, they were not successful in achieving the goals that it was supposed to meet. And I'll talk about why that happened, because I have a slide dedicated to this particular mission. But right after that, it became important to then list the following 4, 5, 6, and 7 that need to also be met. So this has been a learning curve. When we're conducting experiments to look for life, we have realized not just what we did not know, but what we should be. We realized not just what we had to look for, but also what to do when we find something that we're not able to understand what it is. And to be able to further remove room for doubt, we had to ensure detectability. We had to detect the preservation potential, whether it's actually able to preserve the samples, reliability, and the fact whether it is compatible with Earth life or not. So these were the different additional criteria that were added to the list of criteria that an instrument has to be able to complete when it is looking for signs of biological activity. And the most important one towards the end is that biological interpretation must be the last resort hypothesis. You cannot assume it is biological. You can only do that after you have ruled out every single possible explanation, which is non biological. So that's how rigorous this has to be. And this is a screenshot of a table from that same paper, which actually gives an explanation for each of these criteria as to how it needs to be met. And I think it's a great paper which people who are interested in astrobiology should read to be able to understand how intricately well planned an instrument needs to be able to achieve its goals, to be able to confirm whether what it's looking for is life or not life. So to talk about some of the unique challenges that are faced by astrobiology missions, it can be categorized as the following. The first is policy related. Now, missions that look for life have to go through something known as a planetary protection review. Now, what exactly do you mean by planetary protection? Once we found that there is a possibility that the spacecraft that is traveling from Earth and going to a different planet or a moon or wherever into space, if it is not being sterilized, if you haven't killed off all the germs and microorganisms that are on there, there is a possibility that you actually might be carrying these germs with you there and depositing it in that surface environment. And they might be able to live in that environment for long durations of time. And in case there is a second mission that follows your first mission and ends up finding some of that life which you have carried in the first mission, you run the challenge of firstly not being able to confirm whether what you're detecting is life that you have found from here or not. And the second is that that life that is already there from the first mission might actually propagate and start its own little bacterial colony in that harsh environment, by which to say that you've actually carried life from Earth to Mars. So it kind of defeats the purpose of trying to understand what is unique native life that might exist on a different planet or not. So to do this, the United Nations, COSPAR committee has formulated a very strong planetary protection guidelines. And in fact, last year we had an online astrobiology course at Amity University, and there was one class that had been given by Dr. Halisa Hadaji from Harvard Law School, and she's actually been part of the drafting of the planetary protection guidelines as well. And that entire class actually talks about the importance of this. And every planet or moon has its own ranking in terms of the score, in terms of how strict a mission needs to be in terms of conducting astrobiology there or not. And Mars is obviously very high up. And then icy moons of Enceladus and Jupiter and Europa of Jupiter are also quite high up, because these are prime candidate sites for us to look for life. So the planetary protection guidelines are even more strict in terms of the sterilization, the cleanliness, and the rigor with which the assembly needs to take place to ensure that we do not take any contamination, nor do we receive any contamination from the surrounding environment of that planet. So that's something very unique that not all space missions have to fulfill. It's mainly because we have such a goal of looking for life, and we have to ensure your instrument and hardware are able to be within these guidelines. So that's a policy-related challenge. Coming to the environment. Now, of course, space is a harsh environment, and most space missions face their own challenges in terms of temperature, pressure, radiation, and mechanical stresses. But these stresses are even more if you're trying to land on a surface of a planet, if you're trying to land or fly through the atmosphere of another planet, which most of these other space missions relatively do not have to do. They can make their own observations from a distance, right? But for astrobiology, you really need to get up close and personal, where you really need to be in these elements, natural elements of this world, to be able to complete your scientific goals. So the stresses and the challenges are even more harsher for your specific instruments, with which it has to go through. Mission related challenges. Now, astrobiology missions are expensive because they have to go outer into space to these different planets and moons. So, the amount of fuel that is required to travel that far away is also more, and that leads to the mission cost going up. It takes a lot of time to plan out. The instruments are sophisticated even more and need to go through even more amount of testing, and all of that testing and integration time kind of compounds the cost that is required. The number of people involved in these large missions are also high, so therefore also the cost goes up. And missions like Cassini, New Horizons, these have been, their entire mission life can run into more than a decade. So all of that leads to all these excess of resources that need to be accounted for. So it really adds up to why is it so hard to build an astrobiology mission. And lastly, some of the challenges in terms of the instruments. So these instruments have to be really, really smart. They have to be having the ability to analyze unexpected signals or operate in unusual circumstances to be able to do all of these things. You know, where you kind of are preparing for an examination and you don't even know which subject that, I mean, you know what subject it is, but you really do not know to what extent or what depth your questions might be in. So it's the amount of preemptive training and planning and design work and automation that needs to happen is a lot more than some of the other instruments, because they're actually going into ask a question which is not an easy question to ask, which is to understand whether the source of that process, whether it is biological or abiological or abiotic. Secondly, non-destructive analysis techniques. So on Earth, if you had to go around look for whether there is life in a particular environment or not, you pick up the rock, you can break it open, you can see the crumbs of that rock and analyze the powders of it, and then try to break that down and do all of that. Whereas in space, all of that is energy intensive, and also you don't want to break the integrity of your sample. So most of the times you are limited to non-destructive analysis techniques for being able to do this kind of missions. So this is to just kind of summarize the different challenges that are faced when you're kind of doing astrobiology. Now I'll talk quickly about the different missions. So talking about NASA's 1976 mission, which was Viking. It was a well planned out experiment, but had not foreseen the kind of, rather than finding answers, it found even more questions, which in the long term was good for science and it was good for Mars exploration, because we know these things that we have to plan out for. But for that mission, it wasn't so good, because it actually ended up creating further room of doubt, and it was not able to rule out the fact that what it's looking at is actually completely abiological in nature or not. So the experiments at that point of time were rudimentary, because our understanding of the challenge was also not complete. But things have improved even since the early 90s. The Mars exploration program had kind of a renaissance, and through the 90s into the 2000s we've had a series of successful lander and rover missions, down to Curiosity rover, which is still active and was able to confirm that there in fact was flowing liquid water on the surface of Mars. By looking at the geology and the chemicals and the minerals that it had confirmed, it was able to confirm that in the past, Mars was actually a habitable environment. And down to the 2020 rover, which kind of takes the same rover structure and takes it one step further, is a full-fledged astrobiology mission. The primary task of this rover is to look for signs of present or past life and to confirm whether it's able to find proof of biological activity and to collect and store samples. And of course, all of you might have in fact seen the helicopter flight, which was the first ever flight of an aircraft or a helicopter, depending on how you characterize the vehicle, on a different planet outside of our own Earth system. So these are all founding steps into comprehensive surface operations, which will help us further confirm and understand these different planets. And one of the scientists, in fact, Dr. Jennifer Blank, she's also a visiting researcher at our center at Amity. She's been at Amity multiple times over the last few years. And we were actually at Lonar Crater in Maharashtra. And this is the only crater site, one of the only 3 crater sites in the world that is formed in basalt, which is this really hard volcanic rock. And this crater is similar to the crater on Mars where Perseverance rover is, the Jezero crater. And there are several similarities that are drawn between Lonar Crater in Maharashtra and Jezero crater on Mars. And craters are exciting to look for life because you have these water bodies. There's a cavity in the ground and water seeps in, collects over long periods of time. And of course, right now, Jezero is dry, but in the past, it had, as you can see in the lower center image of the crater, you have this delta formation, wherein you have the stream ancient stream that was constantly flowing in and depositing organics and in sediments over a period of time. And that's exactly what the Perseverance rover will be doing. It will be studying the deposition of sediments over a long period of time. And Lonar is an actual active crater site where we can actually study and confirm the habitability potential of these different features of the crater, whether that is the gullies or the delta. And Lunar also has this lake that has formed over a long period of time. And that lake has formed because of these freshwater streams that are constantly flowing into the crater. So all of these are places, analog sites that actually help us understand how can we train our instruments to look for signs of biological activity. The European Mars rover 2022 is also very exciting. I had the privilege and opportunity to do a bit of thermal design work in my PhD time for this rover as well. And it has several subsequent experiments that are also aimed towards looking for life, subsurface exploration. It has a 2-meter drill, which will be the first time ever that such a drill will be used on another planet. And it also has similar objectives to look for life, where it's undertaking these spectroscopic measurements, where it's looking at the bulk, the mineral chemistries, the organics. It's also looking at the subsurface water distribution with its ground penetrating radar. And it also has a hyperspectral microscope on board to do the same. Going further out into the solar system, let's talk about some of the icy moons, right? Now, Cassini was a highly successful mission. It spent a long time observing the Saturn system. It studied the Saturn's rings, understanding how exactly do these rings form, what do they consist of. It had a lot of contribution towards understanding the structure of the Titan moon, which is the only active hydrocarbon-based weather system that we are aware of outside our own Earth system. It had liquid on its surface. It's the only body in our solar system where there's a liquid that is present in the environment in terms of weather patterns and other things happening. So it's a very, very conducive environment for energy exchange to take place, although at a very, very low temperature. Icy moons, for Saturn, we have Enceladus, which is again a very exciting place because the structure is of an icy shell on the outside, but inside it was able to confirm these plumes of water that were coming out from the cracks in the ice. And these were images taken by Cassini as it was flying through these plumes. And it was actually able to confirm that, yes, it could be that there is water that is present inside, and it confirmed that it has a liquid subsurface ocean, and it is kept warm because of the ice that keeps cracking and moving back and forth. So there's friction, and this also happens because of the gravity pull that is exerted by its host planet, which is Saturn. So all of these activities have led to the constant liquid state of the water, and it could be that there is life forms, living organisms that have independently evolved and survived inside these ocean worlds as well. Titan, of course, the surface, some more images of the surface. And these alien weather processes, right? A few years back, we had these images coming back of these very odd, unique, hexagonal shaped polar vortices that had been observed, and they are constantly changing in terms of how they behave. And again, these are different weather systems outside of our own planet's weather system that exist, and they kind of are begging for us to explore them. And we don't even have a complete understanding of what things happen on our own planet. And then when we look in our neighborhood and we find these gigantic gaseous planets, we see complex weather phenomena taking place that we do not have a complete grasp over. Talking about asteroids, some of these other rocky bodies. So Hayabusa did an amazing job by collecting samples from Shogu, which is C-type asteroids. And why are these important and relevant for astrobiology? Well, these are the remnants of the formation of our solar system. We have organics, we have ice, minerals, water that is present. And these are similar elements than what we have in some of the rocky planets in our solar system. And they help us, understanding these molecules help us understand how exactly did our own planet come into be. Where did water come from on Earth? Did it form on its own, or was it actually brought in by asteroids and comets from elsewhere? Comets, again, very, very exciting. These are icy bodies that are constantly orbiting and constantly emitting this trail of water vapor as they approach the sun, and that's what leads to having this kind of structure that floats around. You see that on the top right, this is the mascot of our online astrobiology course called Ketu, which we very fondly have put together. And Rosetta mission was an exciting mission from the European Space Agency, which actually landed on the surface of a comet and was able to confirm so many different things about the active processes that take place on it. And just like asteroids, comets also are a repository of information, kind of like a time capsule waiting to be further explored and holding on to some of those complex organics, those amino acids that could potentially have been the initial building blocks of life that we have on Earth, because you have constantly the source of information that is orbiting and keeps coming in close proximity to our own planet. And at some points of time, because of any direct impacts or constant change of materials that keep taking place between Earth and some of these other planets, it could be that life, in fact, on Earth started because of external interventions from these other objects. This is an exciting clip that was put together by a citizen scientist that is comprised of 25 minutes of videos of images, still images that were kind of stitched together from the surface of the comet. And you see this hazy but grainy image wherein you have these objects, dust grains that are floating around that almost look like snow, but it's actually dust grains that are constantly moving around. Talking about the challenges, now Mars has several exciting places that we are still exploring. We don't have a complete grasp over. And there's a constant debate and discussion and deliberation about where we'd like to land on the surface of Mars. And some of the exciting places that I've highlighted here are these ancient hot spring sites that exist in Columbia Hills, or these deltas and gullies that you find in Jezero Crater. Well, both these places hold their own benefits in terms of helping us understand where life could have started. The other challenge is miniaturization. As you might appreciate, that these are places far, far away. They require a lot of fuel to lift off Earth's gravity and then eventually reach there. Well, at the end of the day, at the end of your rocket, you would only be able to fly a few 100 kilograms worth of stuff onto the surface of Mars, for example. And most of that kilograms would actually be associated or attributed towards the structure, the wheels, the batteries, the power, the panels. So in the end, for the instruments, you just have a few kilograms or tens of kilograms that are left. And that's where the challenge is, because most of the times you have heavy instruments in our labs that are actually needed for doing some of this analysis. For example, the X-ray, fluorescence, or diffraction analyzer. But scientists come up with ways. This is Dr. David Blake at NASA Ames, who was actually able to miniaturize the XRD into a portable instrument called CheMin, which is actually installed on the Mars Science Laboratory. The other challenge, of course, is environment. With Mars and Moon, we know that these are dusty environments. We see how dust covers solar panels, and over a long period of time, limit the operations because you're not able to clean the dust, and therefore the life of the mission is over because you're not able to conduct further experiments. And you also have, for surface missions, the Apollo missions showed us how the lunar regolith actually stuck onto their suits and finds ways to get inside either your spacesuits or inside your rover or your vehicles. And because it's so abrasive, lunar and Mars regolith is a lot more sharper and abrasive because there's been very little amount of weather processes that have taken place, less on Moon, of course, than Mars. And Moon is even more having sharp, angular particles. And these are so destructive over a long period of time that they increase the amount of wear and tear and need special attention. So these are special environmental constraints that come in. Power generation, of course, because you're so far away from Earth, when you're talking about Mars, and even further if you're going towards the icy moons, you're not getting enough solar energy. And NASA has, of course, flown several missions that rely on radioisotope heating, wherein you have energy that is being emitted naturally by this radioactive material, and then that gets transferred and converted into thermoelectric energy, and then you store that, and then you use that energy for either heating up your instruments and ensuring that they don't get too cold, or just converting it into basic electricity for your operations. And then there are other passive methods such as using silica aerogel, but it also has its own disadvantages in which it cannot be used for several astrobiology missions. Lastly, the last two challenges, one is the sample handling, wherein we have faced several challenges by doing either sampling of picking up of soil using scoops, or having drills that can actually drill through the surface. It's a lot easier when you have humans around who can actually pull out a drill in case it gets stuck, and the time response time between what you're seeing is happening and your operations is less. But on a different remote planet, it becomes even harder because a lot of it has to be automated. The drill has to be prepared to drill through an unknown regolith material, which it's not completely sure of. So the drill bit, the type, the configuration, the amount of power needed, everything you're kind of unsure. And at the same time, if it gets stuck, there's several different challenges at play there as well. And if you're doing an astrobiology related drilling, it becomes even more challenging because you want to ensure that you don't contaminate, and contamination and storing the pristineness of your sample is an even bigger challenge. And this is something that I was personally involved with using different kind of sample acquisition methods. And this is one method that I was working on called Planetvac at Honeybee Robotics. And we were mainly looking at using pneumatics, so using forced gases to kind of force up loose, finely grained soil regolith into the lander for further analysis. The last challenge I'd like to talk about is the communications. So communications takes a while, takes time if you're trying to do it from a distant planet or a moon, and more so when you're trying to oversee astrobiology related scientific operations. You really need to have a very short amount of response time. And Curiosity, for example, has different ways in which it can talk directly to Earth, either through Expand directly, or it has an orbiting mission on top like MRO or Mars Odyssey, and then it can relay the information back to Earth. But all of these delays really lead to challenges in terms of how fast you can actually do things on Mars. So to end the talk on my missions, this is I hope some important information that you can take away from it in terms of what do we mean by astrobiology experiments, what are the different types of missions, and what are the challenges that are faced by them. So very quickly, I'll give you an overview on my own journey of how did I go about things, and I'll talk about astrobiology, hopefully within the next 5 to 10 minutes, if I can do that. So things started here in India. I finished my bachelor's at Amity University. I was at IIT Bombay for a year. I moved to the Netherlands for a master's in aerospace engineering and focused on space systems. And I got an opportunity while I was there to work at NASA Ames Research Center, initially for a summer internship, and then eventually as a visiting researcher. And I worked on a couple of space biology experiments on the International Space Station, and that was also my time to get kind of oriented into astrobiology, because a lot of very exciting astrobiology work actually takes place at NASA Ames. It's one of their foremost research departments there. From there, I realized that to further evolve in this discipline, I needed to specialize and get further trained, and moved to Australia on a scholarship to do a PhD at University of New South Wales. I was in Canberra for 4 years. And while I was there, I started working with the NASA Spaceward Bound program, which is a program that allows researchers, students to work together in the field and explore different Mars-like terrain. And that's what I did. And I got the opportunity 2 years back to come back to India and establish this center that we have here. And just to kind of give you an overview of the center in Amity University, Mumbai. So we focus on studying some of these Mars-like environments in Ladakh and Lonar Crater. We are also involved in building microgravity research experiments, for ISRO. And we also undertake a lot of student training and development. So we are a research center. We work with the engineering and the biotechnology school at the university. And we engage with public, and talks such as these are actually part of the work that we do. And we have forged several different partnerships for our different research goals, both internationally and nationally, for conducting some of the work that we're doing. So I'd like to point out this exciting project that we have formulated. So Amity has plans to establish a research station in Ladakh, and as part of this program, we will actually be conducting, starting this year. We were supposed to have a full 5-month season program called the Earth Space Exploration Program, but it'll be kind of condensed into a 2-week program. But the idea is to conduct different science and technology activities. And as part of this Earth Space Exploration Program, we'll be taking in students and training them on different instruments and experiments to be conducted in this Mars-like environment in Ladakh. Now, why Ladakh? Ladakh being a high altitude region, since 2016, we have been exploring it to be an early Mars analog. It has several different environmental parameters and geological features, and hot springs and saline lakes and glaciers that have allowed us to do a lot of important work that is informed Mars exploration. To give you an example, the ExoMars Habit payload, which will be flying to Mars in 2022, was actually field tested in Ladakh. And there are several different teams from NASA, European Space Agency, India, Australia, and we've been working on different kinds of technologies. And some of these technologies that we'd like to further explore, I've highlighted on the right side in terms of communication, teleoperations, suit testing, rover operations, energy and liquid waste storage and treatment. So this is the program that we had put together. Unfortunately, due to COVID this year, we are not able to do a full 5-month program like I mentioned, but there will be a 2-week program in July, 19th July till about 2nd of August. So after my talk, I'll share the link of that as well. And the theme is past, present, and future, wherein we are focusing on exploring Ladakh from the past perspective to understand how Himalayas form. So once you're there, you're able to understand how did these geological processes take place, understand hot springs and how hot springs are important for life to have started. Talking about climate change, how visibly we can see right now the active processes that are taking place there. And then from future perspective, that how can Ladakh help us train for Mars exploration. I've already spoken about Lonar Crater, and hopefully towards the end of this year, we can have another trip to Lonar to conduct some more Mars analog work. And I already mentioned this, that we have been selected for 2 flight experiments on the PS4 platform, and we have submitted some proposals for the Gaganyaan capsule as well, which are currently under review. So that's about it. To kind of give you an overview, we have plans to do work in the low Earth orbit. Down the line, we want to do experiments that can be conducted on the lunar surface and eventually on the Mars surface. So that's all from my side, and sorry for going beyond the time that was given to me, and hopefully we can have some questions now.
Yes, sir. Firstly, I would like to say that thank you very much, sir, for this very enlightening session. And now, yes, of course, we would like to take this time to pick your brains a little more and ask you a few questions that have been submitted by our team members and the audience through the YouTube live chat. So, moving on to the first question, it's a very interesting one. So, it says, how can the findings from astrobiology missions benefit humanity and the life on Earth in general?
That's a very thought-provoking question, because I hope with the current situation that we are faced with the pandemic, we have realized how fragile we all are. And whenever such a thing happens, we all kind of scurry into our houses, and then things get slightly better, and then we go on and we completely forget everything, right? So I talk about how there are certain questions that go beyond countries, regions, localities, societies. This is something that is concerning not just us, but our entire existence, right? So these are existential questions that are kind of waiting to be answered. And several of the technologies, you can answer it in several levels. One is 100 years from now, 200 years from now, what is it exactly that what we would have done would be of consequence to the people who live at that point of time. So if you think from a sustenance point of view, of a humanity sustenance point of view, these are certain questions that will always exist. They existed 2000 years ago. They will exist maybe another 100 years if we haven't answered it. But these are questions that, as creatures that are on this planet who have managed to evolve beyond all these things, they are waiting for us to be explored, right? So that's the first thing. It begs us to answer them. Technologies that come out of space exploration, we have seen how they are relevant. In fact, some of the techniques that many of us are using almost every day, RT-PCR, the different kinds of technologies that are utilized on Earth to understand these harmful organisms. So as soon as biology starts affecting us in a good way or a bad way, it suddenly concerns us, right? But then it also should make us understand that several of these biological techniques, genetic techniques, DNA related techniques, actually happened at a time when humanity in the late 70s was actually thinking about space while it was also developing biology. So a lot of the techniques, so astrobiology started at a time when space exploration was kind of skyrocketing literally, but at the same time, the advances in biology and molecular biology were also going up, right? That's when astrobiology started. And several of the techniques and the scientific fervor with which things were moving forward actually helped each other, complemented each other. And today, many of the organisms that we find living in these extreme environments will and are helping us create medicines, create different ways in which we further understand how living organisms operate, both at a singular cell organism level or a multicellular cell organism level. So people immediately disconnect. The question that you ask me, it's a very innocent question, but at the same time, it has an assumption that astrobiology is out there. How does that concern me? Well, everything is connected in its own ways. And that's exactly what we'd like to point out, that by working on these problems, you're not just might just be addressing the problems that you'll be facing in the near future, but are working on something that will have immense consequence on the existence of humanity by itself.
Yeah, thank you so much, sir. Also, I'd like to mention that during this presentation, we have got a lot of questions on live chat. So thank you, audience, for all these amazing questions. We'd encourage you to ask us more questions. So let's move on to the second question. How is India making its advancements in the field of astrobiology, and what role will privatization play in development of astrobiology sector in India?
That's a great question. India is still at its nascent stages, I'd say. I mean, so I'm part of a community of people who are formulating a society of astrobiologists in the country. And there's been a lot of relevant research happening, which actually is part of astrobiology. For example, there's Dr. Sudha Rajamani at IISER Pune, who is a prebiotic chemist, and she's been an astrobiologist in hiding for the longest time. But finally, she has definitely emerged and is very vocal about the fact that she is doing astrobiology, and we congratulate her on that. And there are other earth scientists who have been exploring niche extreme environments, either in the Antarctic or in the desert arid environments on Earth or in the subsurface. And all of this research actually pertains to astrobiology. So slowly and steadily, definitely in a distributed manner, astrobiology has been happening. What we have done in the last couple of years is we have kind of formalized ourselves into a group of people who are now speaking with ISRO. We've had discussions now for almost 1 year, and we've in fact submitted a roadmap document to them for astrobiology. We've had a couple of committee meetings with them as well. And we are hopeful in the next few years. In fact, Department of Science and Technology in 2016, as part of our Ladakh expedition, had already announced the opening of an astrobiology center at BSIP in Lucknow. So both DST and ISRO in many ways are working on this. And we are hopeful that there will be an astrobiology nationwide program supported directly by the government that will be announced. But meanwhile, at an institute level, definitely there are several institutions that are doing work in this area. That is the first answer to your question. Second part about privatization, it's again very, very exciting, given the fact that we've had almost 40 to 50 new companies that have emerged over the last 10 years in India, in space, in this new space ecosystem. Several of them definitely are having a lot of potential in terms of offering us opportunities to launch. In fact, Skyroot Aerospace and Agnikul, and there are so many different indigenous companies that have come up in the last few years. And it's exciting to see the possibility to perhaps be able to launch independently using these private mechanisms, and conduct, say, microgravity experiments in space, or potentially even launch small satellites that can be sent to Mars or some of these other places as well. So things are very exciting, and I'm hopeful in the next couple of years we might even be seeing a lot more university-led or private-led missions for astrobiology.
Thank you, sir. That's a very thought-provoking answer. So moving on to the next question. So it says, to what extent can we conduct astrobiology research without actually going to space, and what is the significance of field work relative to carrying out experiments in the lab?
That's a very good question. Definitely, you can do a lot of astrobiology work on Earth. In fact, I don't even think if there's been an astrobiologist who's gone to space. Most of the astrobiology work is actually happening here on Earth. It's only robots that have done astrobiology in space. Of course, there have been astronauts on the International Space Station who conduct a lot of space biology work. But field environments. So you have Mars analog environments. We've got Ladakh, we've got Kutch, we've got Lonar Crater, we've got limestone caves, we've got the deep sea ocean hydrothermal vents in the Indian Ocean. There's so many exciting environments which are teeming with organisms, which are having conditions that are similar to conditions that existed in early Mars or present Mars, and even on the icy moons. In the Antarctic regions, we have stations where there is a lot of geothermal activity that is happening, and they have these pristine water ponds and pools that exist there. So these are environments in which you can conduct experiments to further understand, firstly, how do those organisms behave on Earth, and secondly, to be able to test your instruments that eventually you want to send to Mars or to Enceladus or these other places. You can test those environments. And why is it important to do it in the field? Because a field gives you a natural setting. A laboratory is a closed setting wherein you cannot simulate everything. It is good to start your test in a laboratory wherein you can control the experiment and the variables, but eventually you have to move into the field because that gives you a completely natural environment, and that is difficult. It is challenging, but it is very important before you send something into space.
Thank you for your answer, sir. So, moving on to the next question. Would we recognize extraterrestrial life if we found it? Which is us basically asking, like, how much clarity do we have on the fundamental definition of life?
Yeah, so that's again very, very important. And I think in some of my slides, I mentioned about those different criteria that need to be met. And I encourage the person to read more about it. It's still unclear. Life takes to the order of 1 trillion chemical reactions to form. That's been the latest estimate. 1 trillion chemical reactions, and it has to have all happened in some sequence or some order. And we still do not have a complete grasp over it. But based on what we see on Earth and based on what we find on Earth, we have certain patterns and certain processes that we can compare it against. So these are things that help us define whether it is abiotic or biotic, whether the chain of that particular polymer was of certain length or of a certain less length, whether the chirality, if it is left-handed or right-handed. So the organic chemistry, the physical chemistry, there are several different clues and signs that we kind of work towards, but we still do not have a complete grasp over whether it is living or non-living.
Thank you, sir. That's a very interesting answer. So, then moving on to the next question, it says, can a complex organism exist in extreme atmospheric conditions?
So it really depends on how do you define complex. Like if you're talking about multicellular, yes, it can, but it breaks down. So it really depends on what kind of environment you're talking about, what kind of organisms you're talking about. Because you have, if you look at our own oceans, if you look at our own deep sea surface, underwater surface, you find a lot of complex life, multicellular life that lives in very harsh conditions as well. But the challenges that they face and the evolution that they have gone through is, I would say, still less challenging than the kind of challenges and environment and the timelines the single cellular organisms have gone through, which run into the order of billions of years, whereas the multicellular organisms probably hundreds of millions of years, right? So from that logic, we mainly start by looking at some of the single cellular organisms that are relatively the first stage of evolution to start with.
Great. So now we are moving on to the live chats. We have got amazing questions from there as well. So my question is from that I picked from live chat was, can a super gas giant planet like Jupiter support life, according to the knowledge we have on HV about life?
So a gas planet like Jupiter is really, really hot, dense. It is swirling with activity. Potentially, yes, it could, but the fact that it might be able to sustain it for that long duration and how it is getting that energy to do that, and how it is kind of insulated from those temperatures for that long duration of time is hard to say. So you have a lot more favorable environments that you'd like to focus on before we try to look at some of the hottest, because the hottest living organism that we know of is about 150 to 160 degrees Celsius. These are some bacteria that are found in our deep sea hydrothermal vents, whereas the temperatures in Jupiter run into hundreds of degrees, much more than that. So I mean, in theory, yes, you could have an organism that you could perhaps think about, speculate about, but to start with, you would want to start with things that are as close as possible to conditions we have seen on Earth, and then we kind of move steadily away from it.
True.
Thank you, sir. That's a great answer. Another very interesting question that has come up from the live chat asks that could you explain more about the challenges faced due to microgravity while carrying out the scientific experiments in the ISS?
That's very interesting. So I'll give you an example of the experiments I worked on. Microgravity, it depends on what kind of organism you're studying. So for example, for us, so we were looking at fruit flies. Fruit flies are these little flies that you'll find hovering over your overripe bananas or fruits at home, and they are perfect model organisms to study and understand how exactly do these um, insects, how do they uh, react or respond to a different environment. And what we noticed with them was that they didn't move, they just sat in one place, so they chose not to react to it. So many times that is one challenge wherein you are not able to evoke a response out of it, and that is mainly with like fruit flies. But down the line, of course, some of the main challenges we face is getting the samples back, because many times when you do microgravity experiments, and this is less of a scientific challenge, it's more of a programmatic or challenge because of the missions, that there's only so many opportunities to get samples back into the Earth's atmosphere. So many times when we design experiments like the ones we're designing right now at Amity, we are designing non-recoverable experiments wherein we are kind of limited in terms of, because we're not getting the samples back. Once they go into space, the experiment will end there, it will not come back. So you don't have the chance to actually inspect the organism after it has been exposed to gravity. So that limits the amount of observation techniques and chemical assays that you can do, because you're not getting the sample back. So you are limited in terms of what kind of things you can send up that can take all the data and send it back to us electronically, digitally, rather than getting the sample back. But that doesn't completely answer your question. Microgravity, it it is difficult to handle experiments. So like for example on the ISS, life as it is hard for doing a lot of experiments, but many experiments have to be designed in a way that are independent of gravity. So if some design requires things to fall in a certain way or fit in a certain way, it won't behave like that, because things will be kind of floating up and down there. So yeah, I mean, that's kind of to give you a summary of the different kinds of challenges that you might face.
True. Thank you, sir. Another question that we found was, any examples of false positives and how can we recognize them?
False positives happen. So for example, when you're on Mars, you're looking at, like even right now, let's talk about Venus. Uh, we found phosphine, and then suddenly everybody got excited because they were not able to completely rule out the source for it, and that could very well be a false positive. We still do not know, and the reason we do not know is because we're not completely able to rule out what has caused it to happen, production of it to happen.
True.
Thank you, sir. Let's move on to the next question. So, this is a big one. If life is found among one of the planets in our solar system, will it trigger another space race or some sort where countries compete to find life? If so, will it have a positive effect or a detrimental effect on the development of humanity as such in space exploration?
Mhm. Yeah, it's a great thought-provoking question, and I think the answer could be quite personal. I mean, it depends on who you ask. If you were to ask this question to me, I would say it would lead to a revelation in a way that people would have discussions about, just like the first moment when we landed on the moon, and that was a turning moment for a lot of humanity. It would again be a moment where we would not just think about ourselves, but we would start thinking more about space. So I think one thing is safe to say that space would again be a topic of discussion in mainstream. It would not just be something that is discussed when a new launch takes place or a new mission lands on Mars or elsewhere. It would be something that would be discussed more. But depending on the kind of life, I mean, if it's microbial life, there's only a few countries in the world which have the capability to launch these missions. So I think space races are not driven by science; they are driven by politics and geopolitics, really. So I don't think they would, the reason why countries compete with each other is not to do good science and make new discoveries. It is to show that they are able to reach there before anybody else, and that's about it. And that's one of the reasons why the Apollo missions kind of stopped, is because they achieved it. They went there, they put their flag down, and they said, okay, and nobody else is coming, so we plan to go back. So I don't think it will trigger a space race, but it will definitely trigger a movement of countries investing into science and wanting to kind of send more missions out there and make discoveries.
Okay. Thank you, sir. I think we can move on to the last question. So, how realistic is the goal to make human beings a multiplanetary species?
Um, it really depends on how you define that multiplanetary. Like, how do you define it? Does it mean if a astronaut is staying on Mars for 1 year, we become a multiplanetary species, or does it mean taking 100 people and having them spend 10 years? Um, it will happen. It will not happen as soon as many people unfortunately believe, who think it
True.
will happen because of one company that is doing a lot of great things, and their timelines might not line up with what might eventually happen, which is okay, which is absolutely fine. I'm guessing in the next 50 years we might just see, in the generation of you all eventually about to retire, probably we might just be a multiplanetary species if things go along in the same manner in which they have been going on so far.
True. Thank you. Thank you for giving such a wonderful presentation or for answering all the questions raised by our audience. Your years of research, understanding of the subject, and ability to present the subject in such an interesting way really caught our attention. We are grateful for the time and efforts you took to share your knowledge with us today, sir. Thank you. Thank you so much. And we would love to have you back again.
Thank you so much, Kadamri and Vikyat. I'll drop some links for Earth Space Exploration Program and our center website. Those of you who are interested in astrobiology, please reach out to us. We'll be happy to work with you.
Thank you again, sir. And now, on behalf of the entire team, I'm much obliged to thank Dr. Vinu, Faculty Coordinator of Creation Labs and Office of Student Welfare, for making it possible to organize and host such a wonderful event, and Dr. Siddharth Pandey for agreeing to be a part of it, and most importantly, our viewers who have taken their time to join us for this online session. We really hope you had a great time listening and interacting, and we hope to see you again for the next event. Once again, thank you so much, sir.
Thank you. Thank you, Vikyat and Kadamri, for hosting me. And thank you for the entire staff. Thank you.